Hello students, let us learn about nerve cell signaling in this module. I will start with the introduction. Transmission of signals across the synapse or the junction between two nerve cells is called as synaptic signaling. Synaptic signaling is an example of paracrine signaling, which involves the transmission of signals by the nerve cells across the synaptic junction. During the transmission of signal from one nerve cell to another, the sending neuron fires an electrical impulse along the axon, which triggers the release of ligands called neurotransmitters. The neurotransmitters cross the synaptic junction and bind to receptors on the receiving cell and cause change in permeability of ions of the membrane of target nerve cells, which may lead to opening of ion channels and change in electrical potential across the membrane. After the transmission of signals, the neurotransmitters released into the synapses are degraded or taken back up by the sending cell, making them ready for transmission of the next signal. Let us now learn about the neurotransmitters. Nerve to nerve signaling, nerve to muscle and nerve to gland signaling involves the release of chemical neurotransmitters that transmits the signals to the postsynaptic target cell. These neurotransmitters are synthesized in the cytosol of axon terminals and are usually stored in the synaptic vesicles. Common neurotransmitters include acetylcholine, amino acids or derivatives of amino acids and nucleotides such as ATP or their corresponding nucleosides. Some types of the neurons may also secrete a group of signaling molecules called neuropeptides, for example, endorphin, vasopressin, oxytocin and gastrin. Neuropeptides are also stored in vesicles which are different from the vesicles of the neurotransmitters. The release of both neurotransmitters and neuropeptides are triggered by increase in cytosolic calcium ion concentration. In contrast to the neurotransmitters, the neuropeptide transmitters are generally released outside the synaptic zone and their effects are often long-lived, which is in hours to days and diverse. Next is the release of neurotransmitters from synaptic vesicles. The fusion of synaptic vesicles with the plasma membrane to release the neurotransmitters is triggered by a localized rise in the cytosolic calcium ion concentration caused by the conversion or transduction of an action potential into a chemical signal. It leads to membrane depolarization, which leads to opening of voltage-gated calcium ion channels of the plasma membrane adjacent to the synaptic vesicles. The channels permit influx of calcium ions into the cytosol from the extracellular medium, which raises the calcium ion concentration in the region of the cytosol near the synaptic vesicles from less than 0.1 micromolar, which is characteristic of the resting state, to 1 to 100 micromolar. The calcium ions then bind to proteins which connect the synaptic vesicles with the plasma membrane and induce membrane fusion, resulting in exocytosis of the neurotransmitter. The remaining calcium ions are rapidly pumped out of the cell by calcium ion ATPases to lower the cytosolic calcium ion level and return to resting potential again. The synaptic vesicle membranes are rapidly internalized by endocytosis proteins unique to the synaptic vesicles involving clathrin-coated vesicles or non-clathrin-coated vesicles. Now let us take up proteins involved in docking and fusion of synaptic vesicles. The synaptic vesicle membrane contains several types of proteins. The first one is the V-type ATPases, pumps that generate a low intravesicular pH. The second one are the proton-coupled neurotransmitter antiporter, which imports neurotransmitters from the cytosol. The third one is synapsin. It is a fibrous phosphoprotein structurally similar to other cytoskeletal proteins and is localized on the cytosolic surface of all synaptic vesicle membranes. The synapsins are activated by phosphorylation through CAMP dependent and calcium ion calmodulin dependent protein kinases triggered by the rise in cytosolic calcium ion concentration. It leads to the release of synaptic vesicles from the cytoskeleton, increasing the number of vesicles available for fusion with the plasma membrane. The next are the vesicle targeting and fusion proteins. RAB3A, a neuron-specific GTP binding protein located in the membrane of synaptic vesicles, mediate the targeting and fusion of neurotransmitter-filled vesicles at synapses. The next are the calcium ion sensing protein. The cytosolic domain of a synaptic vesicle membrane protein called synaptotagmin contains 4 calcium ion binding sites and is believed to act as the key calcium ion sensing protein that triggers vesicle exocytosis. In a resting nerve cell with low cytosolic calcium ion levels, synaptotagmin binds to a complex of neurexin and syntaxin, which are membrane proteins, facilitating vesicle docking with the membrane but preventing vesicle fusion. However, when cytosolic calcium ion level increases, synaptotagmin binds calcium ion, displacing it from the complex and allowing other proteins to bind and initiate membrane docking or fusion. Next is response of synapses to neurotransmitters. Neurotransmitters can produce excitatory or inhibitory response in a postsynaptic cell. When a neurotransmitter binds to an excitatory receptor on a postsynaptic cell, it may open sodium ions or both sodium ion and potassium ion channels, leading to depolarization of the postsynaptic membrane and generation of action potential. On the other hand, the binding of a neurotransmitter to an inhibitory receptor on the postsynaptic membrane may cause the opening of potassium ion or chloride ion channels, resulting in membrane hyperpolarization, which inhibits the generation of an action potential in the postsynaptic cell. Now let us learn about types of neurotransmitter receptors. The neurotransmitter receptors on the postsynaptic membrane are broadly of 2 classes, that is ligand-gated ion channels and G-protein coupled receptors. Although the synapses containing either type of the receptor can be excitatory or inhibitory, the 2 classes of receptors vary greatly in their speed of response. First, let us take up the ligand-gated ion channels and fast synapses. When a neurotransmitter binds to the exoplasmic domain of a ligand-gated receptor on the postsynaptic membrane, it results in conformational change that leads to fast or rapid opening of the channel in the protein. The opening of the ion channel and movement of ions across the membrane causes change in the membrane potential within 0.1 to 2 milliseconds. The change in the membrane potential may lead to fast excitatory. For example, in case of nicotinic cholinergic receptor, or inhibitory response For example, in case of gamma-aminobutyric acid A or GABA-A receptor. In case of excitatory receptors, binding of a ligand to the receptors results in opening of cation channels, allowing passage of both sodium ion and potassium ions, which leads to rapid depolarization of the postsynaptic membrane and transmission of the action potential. While in case of the inhibitory receptors, the binding of ligand results in opening of chloride ion channels, causing hyperpolarization of the postsynaptic membrane and inhibition of the signal transmission. Next, let us see G protein coupled receptors and slow synapses. In contrast to the fast synapses operating through ligand-gated ion channel receptors, many synapses in the nervous system are slow in response and action of neurotransmitters extends for seconds or minutes. Such synapses are generally mediated by G protein coupled receptor on the postsynaptic cell membrane. The signaling by these synaptic G protein coupled receptors is similar to the signaling by G protein coupled receptors in non-neuronal cells. When a neurotransmitter binds to a G protein coupled receptor on the postsynaptic membrane, it activates a coupled G protein that may either bind directly to an ion channel protein, allowing increase or decrease in ion conductance, or the activated G protein may activate adenylate cyclase or phospholipase C, which triggers an increase in cytosolic CAMP or calcium ion respectively and affects the conductance of ions in a link ion channel. Examples of G protein coupled postsynaptic membrane receptors include receptors for neuropeptides, the rhodopsin receptor for light, and the different olfactory receptors that detect odors. Next, let us take up neurotransmitter acetylcholine. Acetylcholine is a neurotransmitter that is recognized by a diversity of receptors and produces different kinds of responses. The synapses that have receptors for acetylcholine neurotransmitters are called cholinergic synapses. Acetylcholine receptors are of two types, that is nicotinic acetylcholine receptors and muscarinic acetylcholine receptors. Nicotinic acetylcholine receptors are ligand-gated channels for sodium ion and potassium ions, and like nicotine, they cause fast excitatory responses that last for only milliseconds. The muscarinic acetylcholine receptors, on the other hand, are coupled to G protein and, like the mushroom alkaloid muscarine, induce different responses. For example, the binding of acetylcholine to G protein coupled M2 receptor in heart muscles results in hyperpolarization lasting seconds. M1, M3 and M5 receptors activate phospholipase C and M4 subtype inhibits adenylate cyclase. Next, let us learn about termination of neurotransmitter mediated signaling. That neurotransmitter or neuropeptide mediated signaling generally terminates in three ways. The first one is the termination of the signal when the neurotransmitter diffuses away from the synaptic cleft. In the second mechanism, the signal may be terminated if the neurotransmitter is taken up by the presynaptic neuron. For example, signaling by most neurotransmitters like GABA, norepinephrine, dopamine and serotonin is terminated by uptake of the neurotransmitters. In the third mechanism, the neurotransmitter mediated signaling may also terminate when the neurotransmitter is enzymatically degraded. For example, signaling by acetylcholine and neuropeptides is commonly terminated by enzymatic degradation of the signal transmitter. Next is amplification and computation of impulses across synapses. Chemicals at nerve muscle synapses have the potential for signal amplification. For example, an action potential from a presynaptic motor neuron may result in contraction of multiple muscle cells. Another characteristic is signal computation, in which a single neuron, like in the central nervous system, affected simultaneously by signals received at multiple excitatory and inhibitory synapses continuously, averages the signals and determines whether or not to generate an action potential. Let us now learn about intracellular neuronal signal transduction. Intracellular signal transduction in the nervous system generally occurs through three important signal transduction pathways, namely nerve growth factor or tyrosine kinase receptor signaling, long-term depression or LTD, and phosphorylation of tyrosine hydroxylase. First, let us take up nerve growth factor or tyrosine kinase receptor signaling. Nerve growth factor is a member of the neurotrophin growth factor family and is essential for different physiological roles including differentiation, survival, and connectivity of synapses of sympathetic and sensory neurons. Nerve growth factor binds to tyrosine kinase receptor, causing dimerization of tyrosine kinase receptor and activating the intrinsic tyrosine kinase activity of the receptor. The activated receptor then phosphorylates its partner receptor, which triggers the RAS cascade that activates multiple protein kinases. Some of these protein kinases move to the nucleus and activates transcriptional activators. This RAS signaling pathway of nerve growth factor plays a role in inducing and maintaining differentiation of nerve growth factor sensitive neurons. Secondly, the phosphorylated tyrosine kinase receptor TRKA may stimulate phospholipase C, which increases production of inositol triphosphate and diacyl glycerol, which induces release of calcium ions from the endoplasmic reticulum or activates protein kinase C respectively. Phosphorylation of tyrosine kinase A may also activate serine and threonine kinase AKT that inhibit cell death and helps in mediating nerve growth factor dependent survival of sympathetic and sensory neurons. Next is long-term depression. Such type of intracellular signaling is observed during synaptic transmission between parallel fibers and Purkinje cells in the cerebellum. The parallel fibers release the neurotransmitter glutamate to the dendrites of the target Purkinje cells, activating alpha amino 3 5 methyl 4 isoxazole propanoic acid receptor, also known as AMPA receptor, AMPAR, or Quisquelette receptor, which are ligand-gated ion channels. The activation of the receptors results in depolarization of the Purkinje cell for a few milliseconds as well as generation of second messengers. The neurotransmitter glutamate released by parallel fibers also activates G-protein-linked glutamate receptors that activate phospholipase C. which induces the production of second messengers IP3 and diacylglycerol within the Purkinje cell. IP3 causes the release of calcium ions, which produce a form of synaptic plasticity that causes the parallel fiber synapses to become less effective, termed long-term depression. Next is phosphorylation of thyroxine hydroxylase. A third example of intracellular neuron signaling involves phosphorylation of the enzyme tyrosine hydroxylase, which stimulates the synthesis of the catecholamine neurotransmitters, that is dopamine, norepinephrine and epinephrine. Catecholamine synthesis following increased catalytic activity of thyroxine hydroxylase can be stimulated by a number of signals including electrical activity, other neurotransmitters, nerve growth factor and other stimuli that elevate levels of CAMP, calcium ion or diacylglycerol. Now let us see the conclusion. Signal transmission across chemical synapses involves the release of neurotransmitters from the axon terminal of the presynaptic cell into the synaptic cleft, and their binding to receptors on the postsynaptic membrane results in change in the ion permeability and change in membrane potential of the postsynaptic membrane. The binding of neurotransmitters to receptors may result in excitatory or inhibitory response, which may be slow or fast. The receptors may be ligand-gated ion channels or G-protein coupled receptors. Chemical synapses may allow a postsynaptic cell to amplify, modify and compute excitatory and inhibitory signals, and signal termination occurs by enzymatic degradation, reuptake into the presynaptic cell or diffusion. Intracellular transmission of signals in neurons also involves signaling pathways linked to nerve growth factor or tyrosine kinase receptor, long-term depression or phosphorylation of tyrosine hydroxylase. Thank you.